welcome everyone to this NPTEL online certification course on biological process design for wastewater treatment. So, in the previous lectures we have already studied the initial basics of the biological processes that occur, the microbiology, and later on we studied the wastewater characterization, and in particular we studied in detail the COD, BOD, TOC and THOD methods of characterization of wastewater, which are directly useful during the biological process design. Now, we are starting another section within this NPTEL online certification course, and from today onwards we will be considering the modeling of biological processes via various reactions and their kinetics. So, today we will start with the various types of reactions or the which occur during the wastewater treatment inside the wastewater, where the not only the substrate or the pollutants are utilized, also different types of biomass and their growth may occur, and during these reactions various possibilities are occur, and we will study the stoichiometry of these reactions which may occur with oxygen, without oxygen, under anoxic conditions, etc. So, all these reactions today onwards we will be studying in detail for few lectures. So, we will start with the some of the basics of the reactions kinetics in general. So, for microbial growth, the microorganisms or the fermentation reaction or a substrate can always be represented by some chemical reactions, and during this microbial growth or substrate utilization reactions, the substrate and the nutrients and oxygen oblique nitrate are the reactants, whereas the various types of products may be the CO2, ammonia, etc. may be the product, and along with them new type of microorganisms get formed, they are also the product. In general, if we know any of the reaction, so this is a general chemistry that if you know the reaction where A and B are the reactants and C and D are the products. So, under those conditions what we have to do is that first initially we will have to balance the equation. So, what we do is that we try to find out the coefficients of the concentration of the respective reactants and products, and these coefficients are called stoichiometric coefficient. So, and these may be like for the reactions given here, these are alpha 1, alpha 2 and alpha 3, and if we know the stoichiometry, that means these coefficients and the rate of production oblique consumption of any of the other raw material or product which are A, B, C, D. So, if any of the rate of production or consumption of the A, B, C, D values are known, then we can find out the rate of production and consumption of all other species present in the that particular reaction. So, and this particular idea will be useful later on when we study later section. Suppose we know the rate of production of species C, that means which is represented by R subscript C, and its unit may be mass per unit volume per unit time. Then we can calculate the production oblique consumption of A and B and D from the following equation. So, if the mass of the species is expressed in kilomole, that means we are using the molar concentration, then the reaction with respect to Ra can be found out from Rc via this equation where 1 by alpha 2. Similarly, for Rb we can find out using minus alpha 1 by alpha 2. So, we use the ratios of the both B as well as C for finding out these parameters. Similarly, for Rc also we can find out. If the mass is expressed in kg, so this is possible that we have the values known in kg per liter or something like this. So, under those conditions we will have to convert those kg values into the molar units, and for doing this we will have to use the molecular weight. So, under those conditions the above equations get modified as here. So, we can see the initial values are same, but we have to multiply together by the molecular weight of A divided by molecular weight of B. Similarly, for RB we can find out, and similarly for RD also we can find out. So, that means if the rate of production of species C in any unit, either mass per unit, volume per unit time or moles per unit, volume per unit time are known, we can easily find the other values, and this idea will be used later on, because generally we perform the calculation experimentally with respect to rate of production or rate of consumption of one of the key elements, which can easily be measured, and once it can be measured, and if we know the stoichiometry of the reaction, we can find out the rate of production or oblique consumption of all other species via this. Now, here an example is given, that the decomposition reaction of phosphine is given here, and we calculate, it is being asked that calculate the rate of production or consumption of the species pH3 and H2. So, we have to find out the rate of consumption of pH3 and rate of production of hydrogen in kilomole per meter cube per day if the rate of formation of P4 species is 100 kilomole per meter cube per day. So, that means the rate of formation is in kilomole, and we have to find out the rate of production and consumption of all other species. Similarly, in the second part it is being asked that if the rate of production and rate of formation of species P4 is 100 kg, not kilomole, then we have to find out the rate of production and consumption of species pH3 and H2 in kg per meter cube per day. Now, the data given is already given with respect to P4. This is in the first part, this is 100 kilomole per meter cube per day. In the second part, it is kg, 100 kg per meter cube per day. So, we will have to find out the molecular weight of all the species B, C and D, and that has been found here 34, 124, 2. Similarly, stoichiometric coefficients are known from the reaction itself 4, 1 and 6. So, these are written here 4, 1 and 6, and if they are known, then we can use this particular general for any general chemical reaction, not necessarily a fermentation or biochemical reaction. If this is known, we can use this particular type of equation where R A with respect to R C can always be found out. Similarly, R A can be written with respect to R B or R C. So, any of these type of general reactions can be written. So, this will be minus 1 upon, this will be alpha 1 with respect to R B, and this is same as R C minus 1 upon alpha 2 is equal to minus alpha 3 R D. So, this will be known, both these are negative, whereas first one is positive. So, using this reaction we can find out this equation, the rate of production of any other species. So, for the present case, the value of R B is given. Okay. So, sorry, the value of R D has been, R C has been given, and we have to find out for R B and R D. So, we can use this equation. So, from here we can easily see the R B value is equal to R C alpha 1 alpha 2 minus. So, this is this, and thus we can find out this is the equation here, and similarly for R D we can find out using this equation. So, once this is known, we can easily solve, and the rate of formation in the initial case is in kilomole. So, we can easily use this particular type of equation to solve it, and it will come out as minus 600 kilomole per meter cube per day. Remember, we are keeping minus RPH3 because rate of formation is given. So, this is there. Similarly, the rate of formation of species pH3 which is getting consumed is minus this is 109.68 kg per day, and rate of formation of species H2 is this. So, we can use the rate of formation equations for finding out the rate of formation and consumption of all other species. Now, we will go further and study in detail the wastewater treatment reaction stoichiometries. So, during wastewater treatment, a lot of reactions occur depending upon the environment, depending upon the substrate, microorganism, nutrient presence, etc. And there are various types of reactions which are possible that we will study in detail. So, we have to, for better analysis, design, etc., we should find out the stoichiometry of all these reactions. So, to obtain a mathematical description of microbial growth, we need to quantify a relationship between the amount of microorganism produced with respect to the substrate and the nutrients consumed and other products formed during the reaction. So, this is very important for understanding the microbial growth. In this regard, it is required to quantify the stoichiometry of growth process for the various microorganisms under various conditions. So, during microbial growth, there are two types of basic reactions which occur. One category is called anabolism and another is called catabolism. So, in the anabolism, it is the production of cellular material from the carbon source and the mineral elements. So, what is anabolism reaction is the one in which the cellular materials are produced. So, in the microbial biomass, the cellular materials get produced from the carbon source and the mineral elements, and this reaction is called anabolism. The cellular material formed in the anabolic reactions are constituted of proteins, lipids, DNA, RNA, etc., which are all the polymers from which cells are made. Now, production of these polymers is not spontaneous and requires lot of energy. So, this is not easy and this cannot be done without energy requirement. So, that means we require energy from some other reactions. For example, the anabolic condensation relation between the amino acids alanine and glycine to give dipeptide alanine glycine has a positive free energy change delta G of this value at 37 degree centigrade and pH 7, and this reaction can be represented here. We can see the reactions happening between the glycine and the amino acid alanine to give the dipeptide alanine glycine along with water. The energy required for this reaction will always be obtained from the catabolism reaction. So, there are other types of reaction which are classified as catabolism, in which the energy is obtained. Here what we are doing is that we are utilizing the carbon source which contain lot of carbon along with some other elements, maybe a nitrogen containing element, to form the cell walls and other related materials. Now, during this reaction the energy is required. Now, this energy will be obtained from catabolism reaction, which are the intracellular reactions that generate energy, and during this energy process, during this process which are similar to like combustion reactions, the various types of carbon containing compounds in the presence of oxygen, they react and give energy. So, for example, a catabolic reaction is the oxidation of glucose in the presence of oxygen, which has a free energy change like this, and for this reaction the reaction can be represented here, and we can easily see that delta H will be highly positive for this reaction, and this energy will go from this particular reaction to this reaction where the energy will be utilized in the anabolism reactions. Now, in cells, inside the cells, the energy vector is most important energy vector is ATP. ATP, which is adenosine triphosphate. Now, this adenosine triphosphate gets formed during the catabolism energy reaction. So, we have lot of energy sources, the catabolism reaction happens, we have some products which get formed, but this ATP also get formed. This ATP gets transferred into in the anabolism reaction, where this ATP, which is actually the energy source, and this is utilized and we get new microorganisms and product, and during this reaction ADP gets formed, which is transferred back to the catabolism reaction. Now, in cells thus the energy vector is this ATP, and ATP is an energy rich molecule which can be represented by this particular formula C10H16N5O13P3, which is when hydrolyzed gives ADP. ADP is adenosine diphosphate. In place of triphosphate we have diphosphate, okay. And during this reaction the energy is released to the anabolic process which occurs. ATP is generated from ADP back in the catabolic reactions and is converted to ADP again in the anabolic reactions. This whole movement takes place from ATP to ADP and ADP to ATP, vice versa, thus exchanging energy between the catabolism reaction and the anabolism reactions. So, this is the conceptual scheme showing the coupling between anabolism and catabolism, and ATP and ADP are the key molecules which actually help in the coupling process. Now, what we will do is that we will study the anabolism reactions under various conditions in detail. Similarly, later on we will study the catabolism reactions under various conditions in detail, and further later on we will combine both the reactions together to get a combined reaction mechanism under various conditions. So, we will study the anabolism first, catabolism and then the combination reactions. So, the overall anabolic reaction for microorganism can be schematized as some carbon source will be there, some other key elements will be there, we will be having microorganisms which are getting formed, and we have products which are getting released. Now, the empirical formula for microorganism that will be used all throughout will be C5H7O2N. So, this is one of the generic formula which is used in the literature, the same we are using here. Now, what we will be doing is that we will be studying different types of metabolism reaction under aerobic condition, anaerobic condition, using various types of substrate etc., or carbon source, and under various conditions of fermentation, nitrification etc., and then we will study the anabolism stoichiometry. Now, an example with respect to aerobic metabolism of ethanol. So, we are trying to understand starting with aerobic metabolism by heterotrophic microorganism, and we are taking an example of ethanol first. So, for this particular reaction the generic anabolic reaction can be written as that we have ethanol C2H5OH. In if element, the key element which is generally required is nitrogen. So, for nitrogen we are taking ammonia, and oxygen is also required, so that we can get formed this particular microorganism, and we have water which gets released. So, we have this reaction which is written here. Now, in this reaction if you see, the stoichiometric coefficients are written as A, B, C and D, and these values are known. So, how do we determine the stoichiometric coefficients of A, B, C and D? So, for this I will suggest that you can go back, study the class 11, 12th or graduation level courses, and where it is described how to make balances with respect to stoichiometric coefficient equations. So, through balances we can find out the value of A, B, C and D. So, we will be not going in very basic, but somewhere wherever some complications are there we will be discussing, otherwise we will be simply going with respect to the balance equation. Now, the oxidation half reaction for this aerobic metabolism by heterotrophic microorganisms with respect to ethanol can be written here. So, what we do is that we are trying to find out for half reaction, oxidation half reaction, then reduction half reaction. So, in the oxidation half reaction the electrons are getting released, whereas in the reduction half reaction electrons are getting utilized. So, first we write with respect to ethanol ammonia and water, that what is how much amount of electron gets released and how much amount of microorganism getting formed. So, if we write this equation, we can balance this, and the coefficients are written here 2 by 5, 19 by 5, 2 by 5 and 4, and this we do by keeping the value of stoichiometric coefficient of ethanol as 1. So, this is one of the key compound we are understanding. So, we are keeping the stoichiometric coefficient 1 here. Now, similarly, the oxidation reduction half reaction where molecular oxygen is reduced to water is given here. So, oxygen is reduced to water via this particular reaction, and the reaction is written here. Now, for the case of ethanol, we already have 4 electrons for each half reaction. So, we need not multiply together. If the values 4 electron and 4 electron would have been different, so we would have multiplied either of the half reactions so as to balance the electrons on both sides, but this is not required for ethanol. So, overall stoichiometry by just adding together both reactions can be written here. So, this is the stoichiometry of the ethanol with respect to aerobic metabolism by heterotrophic microorganism. Similarly, heterotrophic aerobic metabolism with an organic carbon source, general carbon source, which is CW, HX, OY and NZ. Remember, this formula will be using a lot all throughout our this section. So, under aerobic condition, the reducing agent is generally ammonia, and which is oxidized to nitrate, and this metabolism is called autotropic. So, this is there. So, we have this particular carbon source, this is ammonia, we have water, and we are forming the microorganisms, and we are releasing some amount of electron and some amount of H3O4s, and using some balance we can always write, because the CW, HX, OY and NZ values are given, we can easily balance this relationship using some simple balance conditions which is taught earlier. Similarly, the oxidation half reaction for the reduction of oxygen to water can be written by this, already we have studied. Therefore, combining together overall anabolic reaction for aerobic heterotrophic metabolism can be written like this. For the ethanol case, it was generally written earlier. For the general carbon source, which is written by this, the reaction can be written here. Now, going further, we can similarly find out the anoxic metabolism. Till now, we have studied the aerobic metabolism. Now, we will try to find out the anoxic metabolism of heterotrophic microorganisms. So, for anoxic metabolism of heterotrophic microorganisms, the oxidant which is used is nitrate instead of oxygen. So, remember, we do not have oxygen which is available. So, in the reaction, in place of oxygen we will be using HNO3. So, this is one difference which is there. Now, in the oxidation state of nitrate is plus 5 and it reduces to 0 as nitrate is being reduced to molecular nitrogen. So, we can see that in this reaction, overall reaction will be nitrogen which will be getting formed. So, in the anabolism reaction, which is live in place of oxygen, we have HNO3 and HNO3 is getting reduced to N2. Now, apart from this important difference, stoichiometry is same for aerobic heterotrophic metabolism. The half reaction corresponding to nitrate reduction is same. For reduction reaction, the reaction is different because we do not have oxygen. So, in place of oxygen we have HNO3. Now, HNO3 combines together with electrons and H3O plus to form nitrogen via this particular reaction. Now, this particular reaction plus the oxidation reaction which was given earlier, they are combined together to form this particular reaction which is given here, and we can see here the oxidation half reaction is same as earlier for anaerobic metabolism. So, the anabolic reaction is obtained by combining reactions, so that the number of electrons accepted and removed is the same, and thus we have the substrate which is there, which is our general carbon source. It reacts with ammonia, with nitrate to form the microorganism, which is given generally by this formula, plus nitrogen and the hydrogen. The key thing is how to find out this stoichiometric coefficient, and that can be found out by general balance equations. So, we have got to know two types of aerobic metabolism as well as anoxic metabolism for anabolic reactions. Now, we will further study in detail other types of reactions in whole process, and we will continue further in the next class. Today, we have studied only two basic difference we have found out. There are anabolic reactions, there are catabolic reactions. The anabolic reactions, the cell wall and other things are formed, for which we require carbonaceous source and other elements, but however, for this reaction to occur we require energy, and that energy is obtained from catabolic reactions. So, catabolic reactions actually generate ATP, ATP gets transferred to the anabolic reaction, and then anabolic reactions convert the ATP into ADP and use the energy for their reaction. The ADP further gets transferred to the catabolic reactions, where it again generates ATP, and which again goes back to the anabolic reaction. So, further on we studied few types of anabolic reactions and their stoichiometry. There are many other different types of reactions that occur in the wastewater treatment. Inside the reactor, we will study the anabolic reactions of such anabolic stoichiometry of such reactions in the next class, and further we will study the catabolic reactions also for all such type of reactions. So, we will continue in the next class. Thank you very much.